Good morning and welcome to the GW Epilepsy Grand Rounds. It's my great pleasure to introduce today's speaker, Dr. Ed Dudek, Professor and Vice Chair for Research at the Department of Neurosurgery, University of Utah. Dr. Dudek comes to us by way of a number of prestigious institutions where he trained and where he was faculty, including University of California at Irvine, where he earned his undergraduate degree in biological sciences and PhD. His postdocs were at Irvine and at Columbia. He then accepted a position at Tulane in New Orleans, where he was assistant professor and then became professor. He then moved back to California to UCLA, where he was director of the Center for Developmental Disorders, and to Colorado State as chair of anatomy and neurobiology, and currently University of Utah, where he started as chair of physiology, and he's currently professor and vice chair for research at the Department of Neurosurgery. Throughout his career, he has done a number of exquisite studies that have contributed to our understanding about the pathophysiology of epilepsy in the hippocampus, including the mechanisms of synchronization of seizure activity that makes it possible for seizures to manifest clinically. And these studies have shed light on potential interventions to stop seizures and epilepsy. He has studied extensively how neurons communicate with one another, including via gap junctions and via electric field effect. He studied recurrent excitatory circuitry in the hippocampus and other pathophysiologic signatures of hippocampal epilepsy. He has published numerous papers in major epilepsy and neuroscience journals. Without further ado, Dr. Dudek.
Well, thank you. It's a pleasure to be here. I'm grateful for the chance to talk about our work and to try to put our work in the context of other work in the field. I'll just go through these slides real quick here, I guess, about your epilepsy board review course and disclosures. I do have disclosures to Neurotherapeutics Pharma, which I served as a consultant, but more importantly, Epitel, which is a company that we formed after patenting the miniature telemetry device. And I'll talk more about my conflict there. And let me just say, in terms of learning objectives, I want to talk about cellular mechanisms for both acute seizures and chronic epilepsy, but we'll focus on pediatric epilepsy, particularly developing after neonatal hypoxic ischemic insults. And we'll talk about sort of the principles and controversies in this area. Now, what guides our work here is the issue of neonatal seizures and or brain injury being responsible for acquired epilepsy. That's really at the heart of what I'm going to talk about. I'm going to introduce the work with some experiments that were performed by Shilpa Kadam in collaboration with Andy White and Kevin Staley, and focus then later on work with this miniature telemetry system that was the graduate research of Andrew Zayakevsky in collaboration with Mark Lemkula and John Fisher, who are involved in our company, Jeff Ekstrand, who's a pediatric neurologist, and I'm sort of the advisor of this. Now, I want to remind you of some points about acquired epilepsy. And this is very important because even though I know most of the listeners know a lot about epilepsy, I want to distinguish acquired epilepsy from genetic epilepsy. We're talking specifically about the development of epilepsy after a brain injury, and in particular the occurrence of chronic spontaneous recurrent seizures. That is the outcome measure that I believe we really need to measure as we study this type of disease. An important point is that it requires time, occurs after a latent period from the brain injury to the first clinical seizure. And presumably this depends on an epileptogenic circuit, which could be altered in a variety of ways, and I'll come back to that in a minute. Now, many people view the process of acquired epilepsy sort of like this, a step function. And really, this is how I viewed it, and you'll still see slides that are sort of similar to this, that plot seizure probability or frequency or some measure of epilepsy as a function of time. And what you can see here at the bottom is the occurrence of a brain insult, a brain injury, and then a certain time period, a latent period, which in humans can be anything from weeks up to 10 years. And then after the first clinical seizure, there is a higher probability of having spontaneous recurrent seizures. And that's really what this illustrates in a sort of very, very simplistic sense. Now, another hypothesis for this, and clinically a lot of people, if you were to think about it, would say this is really a more, a better way to think about it, is that it's a continuous function. It's not a step function, as you see on the right. And here what we show is, again, the latent period in both figures, the same axes exactly, but this implies a little bit different process. It implies that from the time of the insult, there's a continuous process that's going on, and that seizure frequency or probability or severity increases with time. And a number of neurologists and a number of basic scientists would say, well, it doesn't always increase with time. And that may be true, but it often does. I mean, there's a lot of clinical evidence to suggest that this occurs. And we can talk about that more later. Now, I want to introduce you to some concepts. If we accept this, this idea of a sigmoid function, a continuous function, this is the same plot as I showed you before. And there are really two kinds of ways to think about acquired epilepsy, and a term that's used a lot now is disease modification. In fact, an RFA just came out from NIH for Epilepsy Center Without Walls based on trying to understand better how we can modify the development of epilepsy. And when we're talking about that, it really comes down to what can we do after the insult that's going to change the epilepsy. So many studies that have been done in the literature in animal models really more accurately reflect insult modification. That is, if you have an insult and you do something right away at that time during the insult and blunt the insult, then you're going to change the epilepsy. But to really study disease modification, you want to look at procedures that you can do after the insult. And I'm going to come back to that in various ways. So the second part of my talk, and the part of the talk about neonatal seizures is to ask the question, what part of the insult is important? Because you can't try to modify the epilepsy without understanding exactly what is going on when you have the insult. And so is it the seizures, is it the brain damage, or is it both? And it's probably a little bit of both. Now, by way of introduction, I want to talk about seizures. And I really spent the early part of my career, as a lot of my colleagues, studying how do you generate seizure-like activity in a brain slice preparation. And at the time, 30 years ago, I thought that was studying epilepsy, and I think we all in the field have evolved to another way to look at things. But if you think about what are the possible mechanisms, and this is a very, very simplistic slide, certainly intrinsic electrical properties, sodium channels, a variety of ion channels, voltage-gated channels, could be important, and there are hypotheses in the field arguing that they are important. More work probably is focused on synaptic receptors and their circuits, so GABA A receptors, AMPA receptors, all have been the target of a lot of studies to determine whether they play an important role in the epileptogenic process. A lot of work, NMDA receptors have been a main target. Now, our group and a lot of other groups have worked on a number of non-synaptic interactions. I'm not going to talk about those, even though I think they're quite important in the generation of seizures. They could be important in epilepsy too. But I'm going to focus more on the synaptic mechanisms. And it should be obvious if you think about it at all that anything that affects seizure susceptibility could be important in epilepsy. So all of those mechanisms could be important for epileptogenesis. Now, an argument that I think is really front and center in the field has been in a debate for decades is the importance of neuronal death. Does the insult have to cause frank brain injury, neuronal death? So it's neuronal death or no neuronal death. And in the spirit of trying to make your research question crisp, this is a place where you would think that we ought to be able to have pretty decisive set of experiments. The neurons die or they don't die. And what I'm going to say to you is it's much more complicated than you might think. And if you think about what are the hypotheses that would invoke neuronal death, one of them is the loss of GABAergic interneurons. So in theory, this is one of the longest standing hypotheses, in theory, you could just have a selective killing of GABAergic interneurons, and that could lead to epilepsy. Now, what you might think, though, is if you killed all the interneurons, you'd have status epilepticus, because it would be just like blocking GABA A receptors, which can cause profound status epilepticus and basically can kill animals. Another way that you could have neuronal death involved in epileptogenesis is that it could be the trigger for synaptic reorganization. So there are a lot of people in the field that believe that if neuronal death occurs, you trigger a developmental process that causes the growth of new circuits, new excitatory circuits, and a lot of work has suggested new inhibitory circuits too. So we talk about sprouting of excitatory axons in the formation of new recurrent excitatory circuits. That's new circuits between excitatory pyramidal cells. This is probably most popularized with what's been called mossy fiber sprouting, and that's sort of a problematic area because many people have studied the sprouting of the dentate granule cells, but actually sprouting occurs in all sorts of other hippocampal pyramidal cells and cortical pyramidal cells. For example, we and others have shown the same thing occurs in the CA1 area. So this sort of loss of interneurons and formation of new excitatory circuits has been demonstrated in many labs, in many models, and it's still debated, but it probably plays an important role in epileptogenesis. Now, just to freshen your memory here, I have two diagrams. So if you look at the top, you can see the blue pyramidal cells. This would sort of emulate the CN1 area of the hippocampus, where all the pyramidal cells in a row, like you would see in the hippocampus, very different than neocortex. You can see the red interneurons, and you can see the concept then is that you lose some of those interneurons. As I'm sure many of you know, interneurons come in many flavors and colors, and there are many different types of them, and they have different susceptibilities to seizures and to brain injury. So one hypothesis then is that this plays a role in epileptogenesis. This is a form of neuronal death. I would emphasize that you could have loss of interneurons, lots of interneurons, and not really detect it because the interneurons are only 5 to 10% of the neuronal population. Most of the neurons are excitatory pyramidal cells. Now, this illustrates the other hypothesis, the increase in recurrent excitatory circuits, and again, would probably be a good diagram of the CA1 area of the hippocampus. So the pyramidal cells are all form a layer, and you can see there is likely to be, we and others have shown, recurrent excitation in the CA1 area, but after an injury, after status epilepticus, after other injuries, you have a formation of more recurrent excitatory circuits. So both of these are hypotheses that either represent neuronal death or are thought to be triggered by neuronal death, and that's how we came to focus on this issue of neuronal death. Now, what really brought me front and center into this was a set of studies that involved pediatric epilepsy from 3 different animal models, where the hypothesis was generated that repetitive seizures in the immature brain, as opposed to the adult brain, in the immature brain lead to acquired epilepsy without neuronal death. It comes from 3 different models, 3 different laboratories. One model, hypoxia-induced neonatal seizures at postnatal day 10, has been argued to lead to epilepsy. Yet another model, hyperthermic seizures, is a model for febrile seizures lasting 30 to 60 minutes at postnatal day 10-12, has been argued to cause epilepsy. And yet another series of studies using the lithium pilocarpine model, status epilepticus at postnatal day 20, which is right about when a rat is weaned, that would lead to epilepsy with no neuronal death. Now, my colleagues, Jeff Ekstrand and Kevin Staley, who I've collaborated with for years, particularly Kevin, wrote a commentary that was meant to be somewhat of a challenge to the hypothesis. After all, hypothesis to be tested. That's the way the scientific process works. We test and try to reject hypotheses. So we wrote a paper arguing that neuronal death was necessary for acquired epileptogenesis in the immature brain, challenging the concepts that had been established by the others. And that was really Francis Jensen, Tally Behrman, and Amy Brooks Kayal. And so what we have is a scenario where a hypothesis, a very interesting hypothesis, has been generated, and can we test that hypothesis? And what it led to is a set of commentaries on epilepsy occurrence and the Journal of AES arguing the two sides, and you can look at those later and evaluate them yourself. Now, the core questions that I want to address here today are, do neonatal seizures alone, by themselves, versus frank brain damage, cause subsequent epilepsy? Now, I want to emphasize that it could be a little of both, right? The seizures and the brain damage. And I certainly want to emphasize that the seizures could exacerbate the brain damage. So, but to try to simplify it, we're just going to make it sort of black and white. Seizures versus brain damage. And then what I want to do also is to address later in my talk how to best identify ongoing brain damage during neonatal hypoxic ischemic encephalopathy. And what we do is, classically in the field, neuroscientists, clinical neuroscientists have used EEG to study the seizures. And what I'm going to try to argue is that there's a lot of classical work that says that background suppression of the EEG is a better measure of whether neuronal death is occurring. Is there brain damage occurring? I'll come to that later. Now, what I want to do is talk about some experiments that were performed by Shilpa Kadam. It was part of her PhD thesis. Shilpa now is at Johns Hopkins. I'm going to reuse this as the introduction to my talk that's going to be to study the neonatal seizures with miniature telemetry. Now, the background here for Shilpa's thesis was she used long-term EEG recording of spontaneous recurrent seizures with a hypoxia ischemia model, the classical Rice-Vanucci model performed at postnatal day 7. And it involves 8% hypoxia after a unilateral carotid occlusion. In these experiments, she implanted 3 bipolar electrodes and continuously recorded EEG with the Data Sciences International Radiotelemetry System, which can go for up to 5 months. So we recorded 3 to 5 months in every animal, starting at either 2 months or 6 months, and I'll show you a diagram on this in a minute. And to her credit, she recorded 97.3% of the time for 3,277 days. So you can think of what she spent her weekends doing. And also this was analyzed both by an automated computer analysis system that Andy White and Kevin Staley wrote. They ran all that analysis. That was done totally independent of Shilpa's blinded human observer analysis and brought into virtually 100% concordance. Now, this is the experimental diagram. Again, by way of introduction, you can see on the upper left, there are 3 channels, 1 through 3. If you look at B, you can see that there's the infarct on the right. Channel 1 was right at the infarct. Channel 2 a bit medial. Channel 3 contralateral and homotopic cortex. And below you can see the diagram of how we did the experiment. So we had a set of controls. There were 10 controls. There were 18 experimental animals, and they were all recorded either from 2 to 7 months or 6 to 11 months. Now, the reason that we started at 2 months is because the DSI telemetry system, which is the competitor, one of the competitors of our company, which is a great system, is big enough, at least it was big enough, that you couldn't really implant it in an animal smaller than 2 months. Not ethically, you just couldn't do that. So we started at 2 months. So one group 2 to 7 months, another group 6 to 11 months. The idea is to have coverage in the whole study for a year and to look at then the development of the epilepsy. And this is kind of what one of the key results. And an important point to think about for any study like this, that the seizures occur in clusters. So this is a plot of the data. And if you just look at the upper A1 graph, what's on the x-axis is 0 to 150. So that's 5 months, 30 times five months of recordings and the seizures per day for every day. And you can see what's very interesting here is that the animals, particularly at the beginning, can go 3 weeks with no seizures whatsoever, then have a seizure. You can see that there, 1 seizure or so at day 21, go another several days, then have 10 seizures in a day, and then several more weeks with no seizures. So that makes it very difficult to study and quantify this, and you really have to have the continuous recording. And you can see also that just by simple observation that the seizures continue to increase over that 5-month period. Also, if you look at A2, A2 is a blow-up. You can see up in A1 over there towards the right where we've expanded it. Seizures can occur in doublets, and they're distinct seizures because even though that looks like they might be all one seizure with a silent period, there's still, many minutes between them. And you can see that the seizures, this sort of illustrates a cluster and how the cluster, what's really happening when you see it on 2 days is it's spanning 1 day going into the next day. Now, one question is, are these really seizures? And my colleagues and I have had a debate with many workers in the field on what's really a seizure. These are not simple spike and wave discharges that you can see in the control animals. These are unequivocal seizures. You can see at the top there a full seizure. On the lower right is a calibration bar of 10 seconds. You can see these are tens of seconds in duration, and you can see the expansion of them in B, C, and D, showing that there's a sort of evolution of the pattern, just like you would expect to see in a real seizure. I emphasize again that not all of these are convulsive. Most of them are. Some are non-convulsive, and particularly early on, the seizures are predominantly non-convulsive. Again, this is Shilpa Kadam's work with Andy White and Kevin Staley in our group. Now, one of the things we wanted to study, and I think this is an important point, is ancillary to what we're discussing now, is epilepsy progressive? And there have been a number of debates about this over the decades in the clinical literature, and there's a lot of perspectives you can take. But these data show pretty clearly when you group all the data together, again, you can see there's age of the animal for the first year and mean number of seizures across animals. You can see that the seizure frequency increases. And the black is the non-convulsive seizures. And you can see there at 3 months, which the 3rd month is where we really could begin to record, that most of the seizures were non-convulsive. And then they become predominantly convulsive. But there are still non-convulsive seizures occurring even as far out as 11 months. I won't talk about B and C. Those are just some mathematical analyses of the data in this data set and another data set. But no matter how you look at the data, when you look at the group data and when you look at the individual data, it's a progressive phenomenon. Now, the really interesting part of this in terms of the hypothesis in neuronal death is when we looked at the data, you could see that we could do the histology and find out was there damage or not. Now, an important point to emphasize here, Shilpa designed these experiments so that roughly half the animals with the Rice-Vanucci model would have damage and half would not, okay? And you can do the Rice-Vanucci method a lot of different ways. And the point here is in 28 animals in which we had 10 sham controls, they have everything but the actual lesion, and this was all blinded, so the person who analyzed the epilepsy did not know the treatment, and then they certainly couldn't know the histology because, the histology was done later after the animal had been euthanized, and the person who did the histology didn't know any of the seizure data. So it was all blinded in all fronts. And the 10 sham controls, they had no epilepsy and no infarct, as you'd expect. But 8 of the 18 animals that were treated with the perinatal HI had no epilepsy and no infarct also. So those animals that appeared to have 2 hours of convulsive seizures never had epilepsy and had no infarct. The 10 of 18 animals that had the epilepsy all had a large infarct. In other words, every animal that did not have epilepsy had no infarct, looked like a control. Every animal that did have epilepsy had a large infarct. So it's virtually 100%. So, as my colleague Kevin Staley has said, no hole, no epilepsy. Now, what does that mean? That suggests, of course, that the seizures themselves didn't really cause the epilepsy, it was the damage. That's what it suggests. But maybe a possibility here is that the HI-treated animals with the infarcts in that study, because we're only looking at them behaviorally and we didn't quantify anything, maybe they had much more frequent and/or intense seizures during the treatment than those without the infarcts. So it's really possible that having the damage exacerbated the acute seizures, and that the seizures are a critical factor. It's more than the damage. So that led to the hypothesis that the acute hypoxia-ischemia-induced seizures were more severe in the rat pups that developed an infarct than those that did not. Now, to test that would mean we'd have to record quantitatively during the 2 hours of the seizures, and preferably also forever afterwards, which is very hard to do with rat pups because the rat pups at P7 still need to be with the dam for another 2 weeks. And for those of you who've worked with rat pups, the one thing that the dam does not like is any rat pup that looks any different than anything else, they tend to disappear. Now, what we did here, though, was to say, how can we study this whole phenomenon of the acute seizures? And you can do that by taking the animals away from the dam for a few hours at a time. They do well with that. So we did a quantitative analysis of the electrographic seizures in this neonatal rodent model, and our goal was to do long-term continuous multi-channel electrographic recordings from all the pups sort of forever, but that's, as I said, not so easy to do. In fact, in our hands anyhow, pretty much impossible to do. But here what we did is record during and after the brain insult, and at least try to quantify during the 2 hours of the insult what happened. This required development of a new technology to get good high-quality electrographic recordings. And let me say, speaking, this is to my conflict, I have no conflict with the earlier work. That was really work done with our competitor, their telemetry system, DSI, which is a great system, but it was not going to be great for recording from the pups. And I wanted to make something better than what we could buy with DSI, at least for certain kinds of things. And so we developed this, and by we I mean my colleague John Fisher, who really invented this, and Mark Limkula, who is a bioengineer who's really done a tremendous job at developing this. And the idea is this. We came up with a new way to make a telemetry system, which you can read about in a paper we published several months ago in the Journal of Neurophysiology. It's a methods paper that describes the technology. But this miniature telemetry device is at this point here a centimeter. It's smaller now. Here's a rat pup that has the device put on its head, and the rat pup can live with the dam and do fine. Now, I have to say sometimes the dam sort of messes it up, but mostly the animals do fine. So, and again, you can see in the lower left there, that's my disclosure for conflict. We have a company. I'm hoping to become a millionaire selling rat telemetry systems. I'm not so sure that's really going to work, but anyhow. So here's our experiment for the new set of studies aimed at trying to understand what happens during the hypoxia-ischemia treatment. Okay? So the animals in this scenario received either hypoxia alone or hypoxia-ischemia. And now we did the hypoxia-ischemia in a way that virtually every animal would have a lesion. So we have hypoxia on the one hand, which causes seizures, or hypoxia-ischemia, which we developed in a manner that every animal would have a lesion. The experimental procedure then is at postnatal day 6, we implant with telemetry, this miniature telemetry device. We give them a day to recover. Then we cauterize the carotid, wait a little while, and then implant, and then give them 8% oxygen for 2 hours and record for that 2-hour period. So in more detail below, you can see the recording protocol, 30 minutes of baseline recording, 2 hours of recording during the hypoxia treatment. And then what we also did is record afterwards, and I'll talk about that more at the end. Now the controls had neither carotid occlusion nor hypoxia, but they were sham surgery animals, okay? They all had the surgery except not the occlusion of the carotid. And just to show you the histology, which was done at the end, you can see, this is from previous work, several labs have shown this with the model, hypoxia alone versus hypoxia ischemia, no obvious infarct versus a large macroscopic infarct. So the way this works is virtually every animal with this protocol that gets a hypoxia alone has what appears to be a normal brain. I'm not saying it is a normal brain, I'm just saying that when you do fluorojade staining, you can see there on the right, on the upper right where it says no infarct, that there's no staining, compared to in C and D, where the hypoxia ischemia, if you look at that closely, certainly with the right light, you can see the lesion with the naked eye. It's a big lesion. And you can see on the right there this sort of watershed kind of pattern of neuronal damage that's characteristic of neonatal hypoxia ischemia. It leads to a large, unmistakable infarct. So here are the recordings from the model. And this is a recording here in A of a typical set of seizures. And let me just say that other groups have recorded now with wired systems. Raoul's group and Kapoor's group have recent publications showing recordings, and so these look similar to those. There are some differences that I think have to do with the bandpass that we are looking at. We have a little bit lower frequency recordings, I think, so we can look at seizures and distinguish them a little bit better. And what you see is there are two types of seizures. One in the alpha band, one in the delta band. And you can see below how they can be separated with simple bandpass filtering. So the 0.4 to 4 hertz delta band is in the red, the alpha band is in the blue, and you can see how that relates to above. So you can pick these two seizure types out and quantify them. And just if you just look at the raw data, which you can see, A, B, and C here are hypoxia alone versus hypoxia ischemia versus an untreated control. And you can see the red arrows to the start of the procedure. And if you look along there, and virtually all of these are seizures, there's little or no artifact in this. And in each one, A, B, and C, underneath the upper trace, which is at a sort of slow time scale, and an expanded time scale, you can see in each case the data, the recordings at a faster time scale. So the expanded recordings, the 1 in each of the traces, A, B, and C, is the baseline. That's the baseline. And then 2 is in the middle of the first hour, near the end of the first hour. And then 3 in each case is the seizures that occur near the end. And what it looks like when you look at hypoxia alone is, if anything, the seizures build up, and they may even build up a little bit towards the end, where the hypoxia ischemia, they start off very similar, but then they sort of fade out. And I mean, that's what it looks like. There's just an N of 1 here, of course, so we need to do a quantitative analysis to really assess whether that's true. This would suggest then that hypoxia ischemia does not exacerbate the seizures. If anything, it does the opposite. Now, is that true? So what we did, by we I mean Andrew and Mark, what they did was to go through all the data from all the animals, blinded, go through and using bandpass filtering, separate out the different chunks of data. So what you can see at the bottom there, red is delta, A is alpha, so those are the different types of seizures in the delta and alpha band, and then green is background. And what we did is we wanted to look at each seizure type individually or together and look at the properties of those. So we went through, and you can see as you go from top to bottom that there are temporal expansions. So at the top, you can see the time bar is 60 seconds. So that cal bar in the lower right shows a minute of recording, and you can see the animals going through the different types of seizures. They can occur together or separate, and went through and just marked each time of the occurrence of those. As you go down lower in A, you can see that it's expanded. The time bar is 20 seconds, and then lower still, you can see you can see a little bit easier. It's a little bit bigger. So you can see the different types of seizures and how we went and parcellated the seizures. Now, this is the result of, as you might expect, a lot of work. So 10 animals in each group, 3 groups, all analyzed every seizure during the 2-hour period. And you can see A and B are all seizures, C is the delta events, the delta seizures, and E the alpha seizures. Basically, A and B, the left and the right, are the same data, just expressed differently. So on the left is the raw data for all animals, all seizures, every seizure, and the red and blue lines illustrate the data set. And to the right, it's the same thing, only it's plotted as a graph. It could be a bar graph, but it's just plotted as a graph to be easier to see, which you can see blue is the hypoxia, red is the hypoxia ischemia, and you can see it looks as if on the blue that, if anything, the seizures increase over time, and then the red, the hypoxia ischemia, sort of follows along at the beginning, but then it falls off with time. Okay? Now, we have analyzed that in terms of total power. We've analyzed it in terms of the total number, a variety of different ways. Here's the bottom line. If you look to the left, we have hypoxia, where blue is the first hour and red is the second hour. Now, this is just an arbitrary separation, first and second hour. I mean, we could have done a lot of different ways. But the first hour, you can see hypoxia and hypoxia ischemia, the 2 blue bars, are pretty similar. I mean, they may be a little, hypoxia ischemia may be a little lower, and I can explain why that is. But the hypoxia from the first to the second hour is a trend towards an increase. It's not a robust effect, but if anything, it's up in the second hour. Contrast that with the hypoxia ischemia, which clearly the hypoxia ischemia group shows a clear decrease in the later part of the second hour. In other words, as the time progresses in the 2-hour hypoxia treatment, where some animals have an occluded carotid and some don't, those without the hypoxia alone animals, if anything, their seizure frequency increases with time. But what happens with hypoxia ischemia, presumably due to a progressive injury, but probably not due totally to neuronal death, the injury leads to a suppression of the seizures. So this is just another way to look at the data, but what we wanted to focus on also was background suppression. So I'll come to this more later, but several groups historically have said in the neonatal seizure field that it's not the seizures, it's the background EEG that's the predictor of is there brain damage occurring. So we looked at this more carefully, following up the work from people like Greg Holmes, who has spoken here before, Holmes and Lombroso, and even before that, Watanabe in Japan had proposed background suppression to be a critical measure of ongoing brain damage. And you can see, again, with blue representing the hypoxia and red hypoxia ischemia, using the parcellation technique that we'd shown you before, isolating the periods of background suppression, you can see that the hypoxia ischemia shows more suppression, clear suppression, whereas that does not occur in the hypoxia alone. So not only do the seizures go down near the end of the hypoxia ischemia, the baseline is suppressed too, which is not really new. That's been intimated in the clinical literature for decades, and I'll come back to that. So to speak to the issue of neuronal loss and is it necessary for acquired pediatric epilepsy, these data argue that we explored this idea of maybe the animals with the infarcts had more frequent or more intense seizures than those without. That's absolutely not true. I mean, the data look pretty clear that if anything, the seizures are suppressed in the animals that have the ongoing injury. And so that argues that the acute HI-induced seizures were less severe, not more severe. Now, I have to have as a caveat, there could be seizures occurring in other places of the brain, and like contralaterally, but my proposal to you is that on the contralateral side, they're just seeing the hypoxia, and that's probably about the same as our hypoxia alone animals. Now, I have about 10 minutes, I think, left, and so I'm going to just go through what happens in the subacute seizures, in the subacute period, looking at seizures and background activity afterwards, because many would argue that it's not the period during the insult, it's the period afterwards that's the most clinically relevant, okay? And I think this is going to be something we want to focus on. It's actually quite a bit harder because it's going to take long-term continuous recording for days and days to really study this. But what we found when we looked at these animals in this model, the subacute seizures were recorded only in the HI animals, and really not very many of them, but there were a few, and none in the hypoxia alone animals. Now, right now, we're thinking that this has to be looked at in more detail. We need to ask the same question not only during the insult, but after the insult. And in some ways, after the insult is more critical than during. And so, in the sense of trying to argue, too, that we've rejected that one hypothesis, we haven't totally rejected it because we need to really come back and look at this period afterwards. And I think that this is going to give us an opportunity to look at what is a more clinically relevant kind of scenario. But one thing that's very interesting, and I really wasn't looking to study this, and it was really the student, Andrew Zajkewski, who really made us look at this more closely, which was this issue of background suppression. So you can see the hypoxia alone animal and the control, when you look at the data off to the right 6 hours after, and you can see the different baselines. There's a lot more activity in the baseline of the control animals than the hypoxia alone. Those with the hypoxia ischemia have a suppressed background. And this is an interesting way to look at it. This is just looking at the power spectral density from 1 to 60 hertz. And if you look at the bottom, what you can see is that the sort of pinkish red is the hypoxia alone, the gray is hypoxia ischemia, and the light blue is the control. And a very interesting point to make here is this. So the hypoxia alone animals had all the seizures, if not more seizures, right? That was the whole point of what I tried to show you. They had lots and lots of seizures. But when you look at power in the EEG from 1 to 60 hertz, this is, all animals, all comers, all time, you can see at different periods afterwards that from the very beginning, 15 minutes is the first 30 minutes, because these are 30-minute epics, that the control and the hypoxia alone sit right on top of each other. They're indistinguishable. There's absolutely no difference whatsoever. It's the hypoxia ischemia that drops down. So you can see the suppression in the EEG, and you can see that it takes days, 96 hours, 4 days, and even at that point in the beta band, you can still see some suppression. So what's an interesting issue here, let me just go back and say, this is a really great potential marker for ongoing brain damage, because what Andrew showed is that you could use as little as 10 minutes, you could use as little as 1 minute at different times, and you can still see the effect. So in other words, the marker of ongoing brain damage is suppression, not the seizures, and you can determine this in very short amounts of time. So let me begin to wrap up here and say that, the summary of our subacute data was that 10 of 10 of the HI animals had lesions, 0 of 10 of the hypoxia alone and the controls had lesions. We detected seizures in 3 of 10 of the HI group and 0 of 10 in the others, but that is sort of 3 and 4 hour time epochs of monitoring. And, over 4 days we had about 6 of those, and I want to just caution us here that we really need to analyze this with continuous recording, which, we need to develop a system that we can record with the dam or separate the pups from the dam. But an important point is it's not the seizures so much as the background suppression. The background suppression is unequivocal. 10 of 10 in the HI animals, 0 of 10 in the controls had background suppression. So to begin to really bring it to a close, the hypoxia alone pups with no infarct had more acute seizures, we don't really know about the subacute period, than the HI animals during the treatment. Like controls, hypoxia alone had no detectable subacute seizures, no background suppression, and no subsequent development of epilepsy. The animals that had the hypoxia ischemia with a clear infarct had fewer acute seizures, more background suppression, and did develop epilepsy. So in terms of my second question that I brought to you, how do we detect ongoing brain damage and what might be a predictor of subsequent epilepsy? It's the background suppression. And I want to thank my colleagues here, Andrew Zajkowski, Mark Lemkula, John Fisher, Jeff Ekstrand, who really helped us develop this telemetry system. And wrap up with our initial core hypothesis was that neuronal loss during perinatal brain insult is necessary. It's not necessary, is necessary for the subsequent development of acquired epilepsy. And our data argue that the hypoxia alone, repetitive seizures of P7, it's not only insufficient to cause epilepsy, but power spectral density analysis really raised the issue of, is there really any damage at all? Now, I'm not saying there's no change in synaptic circuitry, function of receptors, how the animals might learn or anything like that. But in terms of epilepsy, in terms of gross major changes in the EEG, just having 2 hours of seizures at postnatal day 7 does not lead to a big change. And I would say that's very different than what you'd expect to see with an adult. And finally, all this work about background suppression really tracks back to Professor Watanabe, who showed back in 1982. In fact, I'd quote Professor Watanabe's one of his classic papers that in perinatal brain injury, those who developed epilepsy showed more abnormal neurological and electroencephalographic findings than those who did not. The more abnormal the neonatal background EEG, as opposed to the seizures, the more frequently epileptic fits develop later. And this really fits with the idea that you can see neonates that will have seizures for, mini seizures for quite a long time and recover just fine. And he used hypocalcemic seizures as a human equivalent to study that. So again, I have this disclosure. I received grants, I received consultant fees, and I have equity in this company that developed this device. And we believe, not just us, but many others in the field, try to develop devices for humans. We're trying to put together an EEG patch. There are other companies that are trying to monitor, whether it be neonates, children, adults. I think this idea of continuous monitoring, whether it be with our system, any of these other systems, will not only provide insights into studying animal models, but will help us understand people with epilepsy too. So thank you very much.
Thank you very much. We have a couple of questions. First, the power spectral analysis that you showed, the gamma band, which showed the superimposition almost between controls and the hypoxia. Did these epochs include seizures at all?
So the period you're talking about after the hypoxia ischemia, right? Mm-hmm. So they included the whole time. Mm-hmm. So in other words, it would have included seizures, but we did not see any seizures in the hypoxia alone mm-hmm. in those periods. Now again, 4 days we had 6 4-hour periods, so that's a pretty small proportion of the time. But during that time period, even if they had a seizure, let's say it was 3-minute long seizure, that's 30 minutes that we're recording, so it might not be picked up. But so we can't, I mean, all we found in that time period was that the only seizures occurred in 3 of the animals that had hypoxia ischemia. Now, Raoul's recorded that the hypoxia alone animals have seizures in that period too, but we did not see that.
There are certain symptomatic generalized epilepsies that we see in children, in which the EEG shows high voltage spikes all the time, and the ictal pattern consists of suppression. Mm-hmm. So the suppressed EEG is the ictal pattern, mm-hmm. and the interictal one appears
very spiky.
Very spiky, which led certain researchers to hypothesize that the spike itself may be the brain's attempt to prevent the seizure. and when those attempts fail, it's when the seizure becomes clinically manifest. What do you think about this in light of your results?
Let me just say that I know sort of 2 points I would make. One is, particularly looking at the pediatric population and looking at animal models of pediatric epilepsy, genetic or acquired in various ways, different types of injuries, I've really presented a very simplified system. So that's one point to make. I know that in the more animal model literature, particularly with the status epilepticus models and in using in vitro slices, that several workers have argued that spikes might actually suppress seizures. Hard to say. I would argue that Kevin Staley, Andy White, and our group have looked at the question of, are spikes predictive of epilepsy? Okay, so if you just look at the EEG before you start to see seizures, that the spikes correlate pretty well with the generation of epileptic seizures. Now, I have to say, nobody is, to my knowledge, has convincingly shown in an animal model with the types of work that's been done so far, again, I may have missed something, that the spikes themselves predict or block the seizures. Right now, the only relationship that I know of is that in those models, and I think this is going to be the case in Shilpa's data, that every animal that developed epilepsy had spikes beforehand. So right now, I mean, our working hypothesis, and we've seen evidence for this, I think Christophe Bernard's group and others have seen that too, that the spikes are predictive. Again, that's a working hypothesis to be rejected. I mean, we're supported. So, hard to say.
Do you have any way to know, or experimentally to question whether the burst suppression you see has anything to do with the seizures themselves, or if it's more related to the import?
We don't know. Our view, bias if you will, is that it represents a ongoing depolarization or block of synaptic transmission in the affected infarcted tissue in the surrounding area in the so-called penumbra. But we don't really know that, and what happens is, as you saw, that it recovers. I mean, the EEG sort of recovers. So right now, that's one of the open questions. You know, this sort of damage-associated depolarization that's been studied in traumatic brain injury, and the similarity to this is a wide open area. I think that it's kind of been hard to study because, you know, you really need an intact brain, intact animal, and so it's hard to do recordings other than your traditional local field potential recording. It's not been so easy to do, you know, more sophisticated patch clamp electrophysiology, but I think there are techniques available now for us to really go look at that. But I think that suppression is just damage, and it's some combination of depolarization and activation and a block of synaptic transmission. But right now, I think that's mostly speculation as opposed to hard data.
Just to be clear, you were talking about the background suppression in that case,
Mm-hmm.
not burst suppression.
Okay, so by burst suppression, exactly how are you describing?
I don't know. Were you talking about background suppression or burst? Yeah, and you're talking about background. Okay.
Yeah, yeah.
That's kind of my question, which is the background suppression kind of can come in two flavors: longer periods of flatline,
Right.
or smaller amplitude continuous activity. Did you have a?
So when I think of burst suppression, one example is you have ongoing spiking activity and then bursting, and that gets suppressed. And so I'm mostly just talking about background suppression here. I mean, we haven't really analyzed it in the context of what you might traditionally call burst suppression. Okay.
And for your data, for the background in your animals, do they just have a smaller amplitude with continuous activity, or is it sort of more quiet, completely quiet periods with normal activity interspersed?
So to us right now, so far as look like what happens is that your signal to noise improves. I mean, it's what you might have thought of as noise, which is really electrical activity going on, that just gets cleaner. In fact, I won't go back to it, but you can look at the recordings. It's just a very smooth baseline, okay? In fact, you know, what that really says in a technical sense is that when you see what you might think is noise in your EEG recording, that's really electrical activity. And you don't really know that unless you euthanize the animal. But this really looks like a smooth baseline. And yet you'll have seizures come right out of that too. Thank you.